Okay, so welcome to this next video in which we are discussing type 1 activation of endothelial cells. Okay, so we've now discussed one of, we've discussed two of the products of type 1 activation of endothelial cells. So we've seen that when a pro-inflammatory molecule such as histamine activates the G protein coupled receptors, which are specifically GQ coupled receptors, on the basolateral surface of the membrane, and if we're talking about histamine it will be the H1 receptor, then what that leads to is the activation of the GQ pathway, which causes the release of calcium into the cytoplasm of the endothelial cell. This release of calcium does two things. One, it activates cellular phospholipase A2, which then breaks down phosphatidylcholine molecules in the cell membrane into lysophosphatidylcholine and also arachidonic acid. And then that arachidonic acid is used to synthesize firstly prostaglandin H2, and the enzyme which which catalyzes that conversion is cyclooxygenase 1. And then also the prostaglandin H2 is then converted into prostaglandin I2 or prostacyclin by prostaglandin I2 synthase. The prostacyclin is then released by the endothelial cells. Okay. The other effect is that the calcium is going to bind to calmodulin, forming calcium-calmodulin complexes, shown here, and this will activate NOS3 enzymes which are in the endothelial cells, and these NOS3 enzymes will start producing nitric oxide. Okay, and now we've got these two products, and I might just add the other one on as well, so that onto this diagram. We've got prostacyclin or prostaglandin I2 and nitric oxide, and these both have vasorelaxant effects, so they cause relaxation of smooth muscle cells. Now we've discussed that of the three types of blood vessel that we have in our affected tissue, the arterioles, the capillaries, and the venules, only the arterioles actually have smooth muscle surrounding them. So it's going to be the production of prostacyclin and nitric oxide in the arterioles which is most important. So in the arterioles, these molecules will diffuse back to the smooth muscle cells that surround the endothelial cells and will cause relaxation of those smooth muscle cells, which will lead to vasodilatation and increased blood flow to the affected area, leading to redness and heat at the affected inflamed area. Okay, and we also said that maybe you can imagine that the other endothelial cells of the capillary and the venules, if they're chucking out these molecules as well, they'll be chucking them out into the interstitial space, and they may well diffuse back and also aid the relaxation of the arterioles, because they will eventually potentially reach the smooth muscle cells which surround the arterioles and will cause relaxation of them as well. Okay, so you'll get relaxation of the arterioles which supply the affected area, and this will raise blood flow. Okay, now what we want to look at is how type 1 activation can also lead to the contraction of endothelial cells, so that the gaps between neighbouring endothelial cells become wider, and this is particularly going to be important in capillaries here, and also most important in venules. So these gaps between neighbouring endothelial cells, they are going to become wider basically, and they're now going to allow certain proteins and fluid from the blood to move through those gaps and go into the interstitial fluid, or the tissue fluid, and we know that that's where the Staphylococcus aureus that caused all of this is, and those proteins that are going to come out, which is known as an exudate, that's going to help fight the bacterium. Okay, so let's now discuss the contraction of the endothelial cells. Okay, so we've already discussed that when calcium goes up in these endothelial cells, it's going to bind to calmodulin, and you're going to get calcium-calmodulin complexes being formed, okay? And these calcium-calmodulin complexes, as well as activating eNOS within the endothelial cells, are also going to bind and activate an enzyme known as myosin light chain kinase. Okay, so this is myosin light chain kinase. Myosin light chain kinase. And myosin light chain kinase is often abbreviated to MLCK for short. So you take the initials of these 4 words, well, the first letters of these 4 words, and put them together to get MLCK. So what happens is initially the myosin light chain kinase is inactive. So we'll give it a colour. We'll colour it in vivid purple. Initially it's inactive, and then when the calcium calmodulin complexes bind the myosin light chain kinase, that's going to activate the myosin light chain kinase. So here is the calcium calmodulin complex. Here's the alpha helix here. Okay, and then we'll have the 2 calciums bound to it in as red dots. Well, 4 calciums actually. Okay, and now the myosin light chain kinase is going to be activated. And what will it do? Well, it's going to phosphorylate a little protein known as myosin light chain, rather obviously. Okay, so we'll have this little protein known as myosin light chain here, which is often abbreviated to MLC for short. Myosin light chain. Okay, and this is now going to be phosphorylated to have a phosphate group on it. So we'll draw this phosphate group like so. So here's a phosphate group now on myosin light chain. Okay, so this is MLC. Now, usually, this phosphate group will be removed almost instantly, basically. So it will be utterly pointless activating myosin light chain kinase, because as soon as it phosphorylates myosin light chain, what's going to happen is another enzyme is going to dephosphorylate it. And this other enzyme which dephosphorylates myosin light chain is known as myosin light chain phosphatase. Okay, so basically it's utterly pointless activating myosin light chain kinase unless you're going to inactivate myosin light chain phosphatase. And myosin light chain phosphatase is similarly often abbreviated to MLCP for short. Right. Okay, so how are we going to inactivate myosin light chain phosphatase? Well, this is where it starts to get interesting, because this is where we come back to those beta-gamma subunits from right at the beginning. So you remember I told you that when we were looking at the activation of the GQG protein, we just followed the story of the alpha-Q GTP, as you always do when looking at G-protein coupled receptors. You never pay much attention to the beta-gamma subunit. But this is where it gets interesting. In this pathway, the beta-gamma subunit is going to be important, and this is how. Okay, so we're following that story now. So what does the beta-gamma subunit do? It's going to lead to the inactivation of the myosin light chain phosphatase. Now, it's not going to do it directly. Instead, it's going to activate another protein. So the beta-gamma subunit, which I'll draw here. Okay, so this is the beta-gamma subunit from our GQ heterotrimeric G protein. And remember, we don't know what specific beta subunits and which specific gamma subunits are there. So this is just general at the moment. Okay, so here's our beta-gamma subunit, and I'll just highlight the full thing in turquoise. Right, so it's going to activate a protein known as GEF. Okay, GEF. Now, what does this stand for? Well, this stands for, so this protein stands for RAS homology. Oops, that should have been in capitals. RAS is a very important protein, so let's put it all in capitals. RAS homology. Okay, so that's what the rho is for. They took the R from RAS and they took the HO from homology. HO. Okay. And then it's guanine nucleotide exchange factor. Okay, so that's always the case. Whenever you see GEF, it always means guanine nucleotide exchange factor. And basically, this is an enzyme which is going to activate another G protein, but it's not a heterotrimeric G protein this time. Guanine nucleotide exchange factor. Okay, so GEF is for guanine, E for exchange, F for factor. Okay, so this is GEF. And now what this is going to do is it's going to take the protein Rho, or RAS homology protein, okay, And this is a G protein, but it's a monomeric G protein, so it's not a heterotrimeric G protein. It doesn't consist of 3 separate subunits, it's just one protein. Okay, so this is Rho, which, just to remind you, stands for the Ras homology protein, and I'll just move this up a little bit. So this is the Ras homology protein. Okay, and Rho-GEF, or Ras homology guanine nucleotide exchange factor, is going to chop off that GDP. And by the way, this is a G protein, so it has 2 states. It has an off state where it's bound to GDP, and then it has an on state where it's bound to GTP. So Rho-GEF, when it's activated by the beta-gamma subunit, is going to activate the Rho G protein by chopping the guanosine diphosphate off and binding a guanosine triphosphate on instead. So you get activation of the Rho GTP. Okay, now what is Rho GTP going to do? Well, it's going to activate Rho-dependent kinase. Okay, so Ras homology dependent kinase. Okay, and then once you've activated Rho-dependent kinase will then phosphorylate and inactivate myosin light chain phosphatase. So we're going to add an phosphate group onto myosin light chain phosphatase, and adding that phosphate group onto myosin light chain phosphatase is going to stop it from functioning. So, to summarize, when you stimulate this endothelial cell with histamine, or just any ligand which is coupled to a G protein coupled receptor, which is then coupled to GQ, okay, what it's going to lead to is the activation of this Rho-GEF, okay, which is then going to activate Rho, which will activate Rho-dependent kinase, which will inactivate myosin light chain phosphatase. So overall, your ligand has led to myosin light chain phosphatase being inactivated within this endothelial cell. In addition, the other subunit of the GQ protein, the alpha-Q GTP, has already led to calcium release in this endothelial cell, which then binds to calmodulin, and that calcium calmodulin complex then activates myosin light chain phosphatase. So you get an activation of myosin light chain phosphatase and an inactivation of myosin light chain you get an activation of myosin light chain kinase and an inactivation of myosin light chain phosphatase. And that means that you're going to get myosin light chain being phosphorylated now. Now, what does phosphorylated myosin light chain do? Well, this is where things get a little hairy, because it's not known the exact mechanism by which this is going to lead to contraction. We do know that it is the myosin light chain, once it's phosphorylated, that causes contraction, but we don't know the mechanism yet, okay? But we do know what it overall causes. So, let me describe what it causes. You see, if you know anything about smooth muscle cells, you will know that in smooth muscle cells, you phosphorylate myosin light chain, you phosphorylate the myosin light chain, and that leads to the contraction of the smooth muscle cell. However, in smooth muscle cells, you have sarcomeres. You have sarcomere structures within the smooth muscle cell. In addition, the myosin light chain is attached to a myosin heavy chain. Okay, so let me just discuss this. So usually, in a smooth muscle cell, what you would have is you'd have a myosin heavy chain, which is far more famous than the myosin light chain. This is the one with the little head and then the fibrous tail. So this is a myosin heavy chain. Okay, and this often is abbreviated to MHC, at your peril, however, because MHC is often used to mean major histocompatibility complex, and that's what most people would assume you're talking about if you're talking about MHC. But myosin heavy chain is another thing that can be abbreviated to MHC. Okay, and basically you have attached to the myosin heavy chain, or wrapped around it here, you have a myosin light chain. Okay, so this is a myosin light chain, or an MLC. And then when you phosphorylate this myosin light chain in smooth muscle cells, this then leads to the contraction of the sarcomere, and therefore the contraction of the smooth muscle cell. So you might say, isn't that just going to be the same in the endothelial cell? And no, endothelial cells don't have sarcomeres in the same way that smooth muscle cells do. So how MLC being phosphorylated leads to the contraction is a bit more of a mystery, but I'll describe as much as I know. I don't know whether this is the limits of mankind's knowledge, but I think it may well be. Okay, because I got all the information that I'm using from a pretty recent review. Okay, so let's now discuss the way that myosin light chain phosphorylation is going to lead to the contraction of this endothelial cell. Okay, right, so let's go over to the next page. Right, so we've got our endothelial cell here. Okay, and we've already discussed that the endothelial cell will have neighbouring endothelial cells. So let me draw a neighbouring endothelial cell here. So here's this nucleus. And then we've got our neighbouring endothelial cell here. Okay, and for some reason this one looks a lot smaller than our previous one, but never mind. Okay, and we've discussed that between these endothelial cells, what you have is two types of junction. Okay, so this outer one that's closer to the lumen, remember, was called a tight junction. Okay, and this other one that's beneath the tight junction here, this is known as an adherens junction. Now basically what you find, oh no, that's an adherens junction. Nearly put adherens junction. Adherens junction. Right, what you find is that attached to these proteins that are involved in the tight junction and the adherens junction, what you have is actin filament. So let's zoom in on this picture, big style, and draw just those two edges of the endothelial cell. So let me highlight up exactly what I'm doing to make it super clear. So in green here, we've now zoomed this up hugely. Okay, and then in turquoise, here's the edge of this other endothelial cell, and we've zoomed that up hugely. Okay, so we now have these tight junctions, and these are made up of loads of proteins. There's actually, I think, 3 separate protein complexes involved in tight junctions, but for simplicity we'll just have one. Okay, so here's the tight junction, and then the adherens junction is just one protein complex, but you know, there are absolutely loads of proteins involved in the adherens junction as well. Okay, so here's our adherens junction. So basically, in both of these endothelial cells, the proteins that are involved in these tight junctions and these adherens junctions will have cytoplasmic domains, and the cytoplasmic domains are attached to actin filaments. Okay, now actin is an important part of the cytoskeletal structure of the cell. It's in all cells, it's not just in contractile cells such as muscle cells. It's an essential part of the cytoskeleton of all cells. Now actin itself is just a tiny little globular protein. However, you can polymerize it into these great big structures. Let me highlight this. So actin will have in pink here, so we polymerize loads of actin proteins together to make what's known as an actin filament or an actin strand. Okay, and this pink's quickly turning into a horrible sludge. Never mind. Okay, so here we have our actin strand or our actin filament. And these actin filaments are very important parts of the cytoskeletal structure. Okay, and they attach to the cytoplasmic domains of these proteins involved in both the adherens junction and also the tight junction up here. Okay, so this is an actin filament. Now basically, when you activate myosin light chain, okay, by phosphorylating it, then somehow the phosphorylated myosin light chain, and this is the bit where I'm just going to have to say what I know and then leave it. Okay, so the myosin light chain here, with its phosphate group attached, this somehow leads to these actin filaments sort of contracting and pulling the membrane basically back, pulling the whole thing back, So these actin filaments contract in response to the myosin light chain that's phosphorylated. That's the information that I've got. Something about this myosin light chain causes these two cells to contract back, and as you can see, when these actin filaments pull, what's going to happen? The membrane is going to be pulled backwards, and the same thing will be happening in this turquoise cell. You'll have actin filaments over here, and they'll be pulling this way basically, and that will mean that this membrane goes this way. So you're going to pull the tight junctions and the adherens junctions apart. These protein complexes are going to be pulled apart, okay? And it's going to open up a gap in between the two endothelial cells, okay? And we'll discuss the significance of this gap in the next video.